Namaskar. Today we will study immunosuppressants. So the drugs which suppress the immune system are called as immunosuppressants. So to before study the immunosuppressants, let us have a recap on the immunity. Immunity, it is the natural defense mechanism of our body, and it can be natural immunity or acquired immunity or adaptive immunity, and that adaptive immunity can be either naturally acquired or artificially acquired, and it can also be divided into active immunity and passive immunity. And the natural immunity can be at the level of individuals, at the level of species, at the level of race. And there are three lines of defense. The first line is the physical or surface barrier like skin, mucous membrane, body fluids like tear, saliva, urine. Then if physical barrier is not sufficient, then comes this non-specific response, which includes the vascular events, which consists of vasodilatation and increase in vascular permeability. There is accumulation of the exudate containing the inflammatory mediators. Then there is also cellular events in the non-specific response, which include the blood cells and other cells. If non-specific response is not sufficient, then comes the specific response. So the physical barriers and the non-specific response provide the natural or innate immunity, whereas the specific response provides the acquired immunity. So in the specific response, there is role of the T lymphocytes. So the antigen, first it is processed and is presented by the antigen presenting cell in the form of major histocompatibility complex MHC, which may be either MHC1 or MHC2. The T lymphocytes on their surface have receptors called cluster of differentiation or CD. Then these cluster of differentiation recognize the major histocompatibility complex of the antigen presenting cell, and there will be a response. So CD4 recognizes MHC2 and CD8 recognizes MHC1. And in response to that interaction, there will be specific immune response which provides us the acquired immunity. And let us see here, when the antigen presenting cell on its surface shows major histocompatibility complex 2, T lymphocytes with CD4 recognize them, and there will be autocrine action of interleukin 2, which will differentiate them into T helper NOD cells, which again can be differentiated into T helper 2 cells or T helper 1 cells or T helper 17 cells or regulatory T cells. T helper 2 cells then differentiated into B lymphocytes, which provides us the humoral immunity through antibodies. And the T helper 1 cells, T helper 17 cells, T regulatory cells, all of them release the interleukins, the cytokines, and have different actions. And when major histocompatibility complex 1 is expressed, it is recognized by the CD8 T lymphocytes, which are differentiated into cytotoxic T cells, which kill the virus infected cell or cancer cells. So when we say the specific immune response, there is the role of the cytokines, and the major cytokine is the interleukin 2. And when there is specific immune response and there is overactivation of this immune response, we need immunosuppressants. So for example, when there is organ transplantation, so there will be activation of the immune system, and we need to suppress the immune system. In that case we need immunosuppressants. So immunosuppressants, we can classify them like this: inhibit lymphocyte gene expression or cytokine gene expression. Glucocorticoids, they inhibit the expression of cytokines, cytokine genes like interleukins. In addition, they also suppress the COX gene and inhibit the release of prostaglandins. And in this way, glucocorticoids like dexamethasone, methylprednisolone can act as immunosuppressant. Then second group of drugs which inhibit lymphocyte activation and proliferation by inhibiting interleukin 2 production or action. So they inhibit the lymphocyte signaling. So interleukin 2, as we already discussed, that interleukin 2 has a major role. And we can see here interleukin 2 has autocrine action and how this autocrine action occurs. So this interleukin 2 is produced when there is interaction between, say, T lymphocyte. So this is a T lymphocyte. This is T lymphocyte, and there is expression of CD4, cluster of differentiation 4, and the antigen presenting cell expresses major histocompatibility complex 2, and when there is interaction between the major histocompatibility complex 2 and CD4, intracellular calcium increases, calcium binds with calcium calmodulin, and calcium calmodulin activates the calcineurin, and this calcineurin can activate this nuclear factor activated T cells, So NF80 phosphate, there will be dephosphorylation to the cytosolic component, NF80C, this is cytosolic component, and N is the nuclear component. So calcineurin activates this nuclear factor for activated T cells, and the cytosolic component and nuclear component together, they induce the cytokine genes, and because of that there will be transcription and formation of interleukin 2 messenger RNA, which will form interleukin 2. So interleukin 2 production occurs in response to the CD4 and MSC2 interaction, and calcineurin also activates octamer activating protein. So OAP phosphate, there will be dephosphorylation into OAP, and this OAP causes the induces the formation of production of transcription factors, and which help in the induction of cytokine genes. Now this interleukin 2 generated acts on the T lymphocytes. So this is autocrine action. So this is autocrine action because the interleukin 2 produced again released from the T lymphocytes, and they again act on the same cells, and interleukin 2 activates the cyclin dependent kinases, which helps in the progress of cell cycle from G1 to S phase in the cell cycle, which will induce the cell division, and there will be T cell proliferation. And mammalian target of rapamycin, this activates the cyclin dependent kinases. So there are some cytosolic components like cyclophilin and FKBP12. This is so called because the drug tacrolimus is initially termed as FK506, and FK binding protein, FKBP is FK binding protein. So cyclophilin and FKBP are the two proteins which inhibit calcineurin. So the immunosuppressants, they either bind to cyclophilin or FKBP and inhibit calcineurin. So cyclosporin binds to cyclophilin, inhibits calcineurin. Tacrolimus binds to FKBP12 and inhibits calcineurin. Sirolimus inhibits FKBP12 but it inhibits this complex, sirolimus and FKBP complex inhibits mammalian target of rapamycin, and therefore they inhibit this autocrine action of interleukin. So in this way we can say now that these drugs can be used as immunosuppressants. So drugs which inhibit interleukin 2 production or action by inhibiting calcineurin. So drugs binding cycloferin, example cyclosporin, drugs binding FKBP, example tacrolimus. Then the second group is drugs which inhibits the mammalian target of rapamycin, example sirolimus, everolimus. Sirolimus is also known as rapamycin, so this target is called mammalian target of rapamycin. So sirolimus first binds to the FKBP, then it inhibits the mammalian target of rapamycin. Then we can have certain anti-cancer drugs which are cytotoxic agents. They, because of the cytotoxic agents action, they inhibit the lymphocyte proliferation, and they include anti-metabolites like purine analogs, pyrimidine analogs, and folate antagonists or folate analogs. Because in the DNA we have the bases, so adenine and guanine are the purine bases, and cytosine, thiamine, uracil are the pyrimidine bases. So uracil is present in RNA, so these are the pyrimidine bases. So we can have anti-metabolites which can be either analogs of purine or pyrimidine. Then there is during the synthesis of DNA, deoxyuridine monophosphate is converted to deoxythymidine monophosphate. Then there will be DNA synthesis. So in this step there is the enzyme called thiamidylate synthetase, and this enzyme requires a coenzyme, that is tetrahydrofolate. Tetrahydrofolate acts as the coenzyme here for this enzyme. So we can use the folate antagonist or folate analog, that is methotrexate, which will inhibit this step. So by inhibiting the DNA synthesis, they can act as cytotoxic agents, and they inhibit lymphocyte proliferation. Similarly, the alkylating agents, they also inhibit the DNA synthesis by alkylating the guanine base. N7 of guanine is alkylated. So there will be cross-linking, interstrand and intrastrand cross-linking will be there, and that will also cause apoptosis, and there will be cytotoxicity. So in this way, these drugs can also act as immunosuppressants. Then we can use the antibodies against specific immune cell molecules, where we can have either polyclonal antibodies or we can have the monoclonal antibodies. The polyclonal antibodies are the antibodies that are secreted by different B cell lineages, whereas monoclonal antibodies come from a single cell lineage. So this is the difference, and to prepare the polyclonal antibodies, we need to inject a specific antigen to the lab animals like rabbits, goats, etc. The animal is immunized repeatedly to obtain the higher titers of antibodies. And in case of monoclonal antibodies, the immunogen is first injected to a host animal, that is mouse. Then after the immune response by the immunogen, the B cells from the spleen are extracted and fused with myeloma cells, that is the cancer B cells, and a hybridoma cell line is produced. So here a hybridoma cell line is produced, so that the monoclonal antibodies, that means a single source will be there, will be obtained. So a specific clone number is assigned to each hybridoma cell line, so that a particular antibody and specific epitope to which it binds can be identified. And mainly when we say antibody therapy, these monoclonal antibodies are used. And four major monoclonal antibody types have been developed. They are murine, chimeric, humanized, and human antibodies. And the initial therapeutic antibodies were murine antibodies, and they were represented by the suffix OMAB, O-M-A-B, OMAB. And then the murine antibodies were prepared by this hybridoma technology, and that is we called it as chimeric antibodies. The murine antibodies then replaced by the chimeric antibodies. Then third generation is the humanized antibodies. Okay, so the final one, the human antibodies, that they use the technology of DNA recombinant technology. So we have different monoclonal antibodies which can target specific cytokines. For example, bevacizumab targets CD52, daclizumab targets interleukin 2, then certolizumab targets TNF alpha, tocilizumab targets interleukin 6, adalimumab. So human antibodies are represented by mumab, and humanized by jumab, and chimeric by schemab. So in this way all these immunosuppressants can be used. And then some other drugs are there, like TNF alpha inhibitor etanercept, interleukin 1 inhibitor anakinra, then specific antibody against D antigen, that is RHOD immunoglobulin. And uses of immunosuppressants. Immunosuppressants can be used in organ transplantation to suppress the rejection of transplanted organs. Drugs like cyclosporin, tacrolimus, sirolimus are used. Then to suppress the graft versus host disease following bone marrow transplantation, we can use drugs like cyclosporin. We can also use for immunosuppressants like RHD immunoglobulin for selective immunosuppression, prevention of rhesus hemolytic disease of the newborn can be done by using this RH immunoglobulin. Then the treatment of autoimmune diseases like acute glomerulonephritis, idiopathic thrombocytic purpura, autoimmune hemolytic anemia, myasthenia gravis. In these cases also we can use immunosuppressants. Then immunosuppressants are also used in severe inflammatory bowel disease like ulcerative colitis and Crohn's disease. Cyclosporin can be used in ulcerative colitis, and infliximab can be used in Crohn's disease. Then in case of severe skin diseases like atopic eczema, severe plaque psoriasis, we can also use immunosuppressants. Tacrolimus, pimecrolimus can be used for atopic eczema which are uncontrolled by topical corticosteroids or glucocorticoids, and for very severe plaque psoriasis which has failed to respond to methotrexate or cyclosporin, we can use drugs like etanercept, infliximab. Then another use is arthritis and related diseases like rheumatoid arthritis, psoriatic arthritis, ankylosing spondylitis, juvenile arthritis. So the disease modifying anti-rheumatic drugs can be used in these cases like methotrexate, leflunomide, cyclosporin. And when these drugs like methotrexate and other disease modifying anti-rheumatic drugs do not respond, then we can use the cytokine modulators like adalimumab, etanercept, infliximab, etc. So this is all about immunosuppressants. Thank you.